Chapter 17, Lesson 2, we are moving on to evolution is genetic change. So we're going to be kind of picking up where we left off from Lesson 1, where we were looking at single gene and polygenic traits and how we graph those. We're going to start by talking about how natural selection can act on both of those. Um, and then we're also going to look at what's called genetic drift, which is one way that evolution can occur. You can have a change in a population. And then just the last thing we're going to finish up with is genetic equilibrium and how we can tell if a population is evolving and the factors that lead to evolution in a population. Okay, so let's start with how natural selection works and look at how natural selection will affect single gene and polygenic traits because it affects them differently. So remember, natural selection is going to affect the phenotype directly. It affects the phenotype. So because natural selection, excuse me, natural selection can change the allele frequency. So when it changes the allele frequency, it's going to change the phenotype frequency. So how often we see a particular trait. So this could be caused by a mutation. So a mutation that might affect the fitness of one individual over another could change the phenotypic ratio. But then again, mutations might not affect fitness. So they wouldn't have any effect on natural selection, or they wouldn't be affected by natural selection. So if it doesn't affect the fitness of the individuals, it's not going to have any change. It's not going to have any dramatic effect on the phenotype ratio. So that's a single gene trait. So when you have like two versions of the trait, right, you might have like black fur versus white fur or something like that. And one of those might be more suited to the environment at the time. And then you could have a mutation that produces brown fur or something. And if brown fur doesn't make that individual any more or less fit, then it doesn't really matter. You probably wouldn't see a whole lot of it. If it makes the individuals more fit for some reason, then the brown fur trait might become more popular. Or more popular is not the right word, more common. On a polygenic trait, natural selection will act in one of three ways. And so it just depends on which type is going to be more fit. Okay, which form or which end of the spectrum. So remember, polygenic traits form a bell curve. Okay, so polygenic traits, you end up with like either extreme and then everywhere in between. So we're going to look at some examples. As you do your homework and whatnot, you can feel free to like make up something as long as it sort of kind of makes sense. All right, so the first type of natural selection on a polygenic trait is called directional. So directional selection means that individuals at one end of the curve are more suited to the environment than individuals at the other end. So this dotted line is our original population. And let's say that this is those finches we've talked about a couple of times, where the larger your beak size, right, the more you were able to kind of branch out and eat other types of food. Okay. And then the smaller beaked individuals really couldn't do that. So they all died, right? So that pushes um, our our curve here to the right, okay, towards larger and larger beak sizes. So the range shifts with directional selection, like literally moves the whole thing in a single direction. Okay, so the range shifts and so does the average. So the average is like right in here, right? So the average is going to shift and so does the whole graph. That's directional selection. Stabilizing selection is where it is better to be in the middle than at either end. One example of this is birth weight. So very, very tiny babies don't do really well, but also really big babies don't do really well. So we have this narrowing of the curve, selecting against very low birth weight or very high birth weight. So it keeps our average in the middle, keeps our average in the same place, um, and creates a very narrow curve. The third type of selection is disruptive, which is where it's not very favorable to be that intermediate form. Okay, that intermediate type is acted against, and they have actually less fitness. Um, and so being at either end is better suited. And so it ends up creating two distinct phenotypes or two distinct groups. I can't off the top of my head think of an example of this, but I will eventually. It'll come to me. There should be at least one example um, in your book. Okay. All right. Genetic drift. There's two types of genetic drift. Either one is leading to a dramatic change in the population. So there's bottleneck and there's founder. So genetic drift means that we have a random change to the allele frequency. Some series of chance happenings have caused one version of the allele to become more or less common. So here we have an example of these beetles, and somebody comes in and smashes a few of those beetles. Well, that's going to change your allele frequency. Okay, because now instead of having three of these green ones, we only have two. Or sorry, we only have one. Two of them died. Okay, so a genetic bottleneck is where randomly something has happened in our environment to drastically reduce the population size. And so when something comes through and randomly affects like a wildfire, perhaps, or a hurricane or some sort of natural disaster that comes through and it kills a whole bunch of the population, um, it would be a very random change. Okay, so it doesn't really have anything to do with being more or less fit. It's just some of us died and some of us got to survive. Okay, so a bottleneck effect dramatically reduces the population size, which can, because it's random, change the population's genetic diversity. Okay, it will change the allele frequency from the original population. So um, if so, here we have these blue and yellow beads representing the original population, and then randomly we shake some out to represent the bottleneck event. Um, well, that's not that we don't have the same allele frequency showing up in our new generation, the surviving individuals. So as they reproduce, we end up with a population that has a totally different allele frequency than the original just because of a random event. Okay. Some chance thing that came through, and these are the individuals, the surviving individuals, it's totally random rather than like those who are most suited to the environment. Founder effect is kind of similar. Founder effect happens when you have a small group that colonizes a new area. So the founders could have very different alleles compared to the main population. So you get this migration of a small subgroup, which could then totally change the allele frequency compared to the original population. Okay, so again, you have just this random change because it's not usually like based on anything genetic that we would have this group go. Um, so you end up with different alleles just kind of by chance because those were the ones that happened to be transplanted to that new area. Okay, so we're going to discuss now for the rest of class, or the rest of this lesson, we're looking at comparing evolution to genetic equilibrium and how do we really define evolution. So genetic equilibrium is when the population is not evolving because the allele frequency is not changing. Um, so because with sexual reproduction, we can shuffle our genes around, right? We can have lots of different combinations of genes. that doesn't necessarily change the allele frequency. That's one of the misconceptions, is that in sexually reproducing organisms, you can't have genetic equilibrium because you have this constant shuffling of genes, but that's not true. Um, you can still maintain the allele frequency, so you would still be in genetic equilibrium. Genetic equilibrium means that you're not evolving. Okay. What we One way that we test for this, okay, is the Hardy-Weinberg principle. This is a prediction of the allele frequency and whether or not a population is evolving. Okay, so kind of like we use the Punnett squares to predict outcomes for genetic crosses, we can use Hardy-Weinberg to predict whether or not the allele frequency is changing. Okay, so we use this for dominant and recessive alleles. So P represents the dominant allele, the frequency of the dominant allele. Q represents the frequency of the recessive allele. And so we have these two equations. You're not actually expected to apply them, but these two equations that let us tell if the population is evolving or not. So we would use these to plug in our values. And if our values that we predict match what we actually see, then the population is not evolving. Okay, and so I think there's an example. Okay. So here we're looking at some trait. The dominant trait is 40%, and the recessive trait is 60% of the allele frequency in the population. Okay. So if the dominant trait, the allele for the dominant trait is 40%, then P is equal to 0.4, and Q then is equal to 0.6. So the chance of being homozygous dominant in this particular population would be 16%. Okay, the chance of being homozygous recessive is 36%. And then the chance of being heterozygous is 48%. Okay, so if we go back, they're using this equation here. So P squared plus 2PQ plus Q squared. So this equation is predicting allele, sort of the probability of each homozygous dominant, right here, homozygous dominant, heterozygous, and homozygous recessive. P plus Q here is just the allele frequencies. So if we have our population and we know that 16% of them should be homozygous dominant and 36% should be homozygous recessive and about 48% should be heterozygous. If we actually look at our population and we see very different numbers, then we would definitely be able to say microevolution is occurring. If the numbers match pretty closely, then there's probably not microevolution occurring. Okay, so that's how we use this as a prediction. Okay, so based on the allele frequencies, we can calculate what phenotypes we should see, the percentages of each phenotype. And if we don't see those percentages, then we probably have some microevolution. There are 5 conditions that can disturb genetic equilibrium. We usually have at least one of these happening in most populations. So most of the time we have microevolution occurring, at least somewhat, in almost every species. So the 5 conditions that will disturb genetic equilibrium are non-random mating, having a small population, immigration or emigration, so individuals coming in or going out, mutations, and natural selection. So non-random mating would mean that there is some sort of sexual selection, that individuals are selecting their mates based on a trait. So like being the prettiest, or having the best coloring, or being the biggest or the strongest, or whatever, that there's some criteria that we use to select mates. And that's actually very common amongst most organisms that, so for instance, like peacocks, right? The males are really colorful. So that actually shows the more colorful they are, the prettier they are, that indicates that they have better genes. And so the females tend to select those males that have better genes because it means their offspring will be stronger. Okay. So um, the genes that are kind of leading to the prettier peacocks or whatever it is that those particular organisms are looking for in their mates, those genes are not going to be in equilibrium. Okay. Having a small population size. So genetic drift is going to have a greater effect in small populations. So if you have a very small population and something drastically comes in and reduces your population size, that's going to have a much stronger effect than if you have a very large population. Okay? Because if you're already a small population to begin with, you already don't have a whole lot of alleles. And if we drastically reduce the number that you have, well, now you're really changed. Okay, now your allele frequency is very different. Immigration or emigration, um, that's going to either introduce new alleles when you have new individuals come in that introduces new alleles, and then individuals that leave obviously take their alleles with them. So we're going to have a change in the allele frequency just by individuals coming in or going out. Mutations can introduce new alleles, and that could change the allele frequency. And then natural selection um, can disrupt genetic equilibrium because if one version is better than another, better suited to the environment than another, then that trait is going to show up. The better suited trait is going to show up more often because those individuals get to survive more and reproduce. So we can change the allele frequency that way. So those are the 5 conditions that would disrupt natural selection. Okay, so disrupt genetic equilibrium. So to maintain genetic equilibrium, you would want to have no natural selection, no mutations, nobody's leaving or coming in. You'd have a large population and random mating. Okay, that would maintain genetic equilibrium because you don't have any forces in effect that are changing the allele frequencies. Okay, so anything that would cause changes to the allele frequencies that would favor one version over another, those are going to cause the genetic equilibrium to shift. Okay. All right, so that was lesson 2. We have one more lesson in this chapter before your test. So I would recommend you start on your reviews.